All right. Hey, everyone. My name is David Barfknecht. I'm a PhD student at SIU, and this is actually my second time speaking for the symposium. I'm happy to be back. So this is actually a separate project from my master's research. Now I'm into this stuff, which is a fellowship project looking at the interactive effects of disturbance that drive species composition and dispersion in a secondary successional field. So something about community assembly is this idea that local species are determined by both biotic and abiotic filters as are determined by the landscape. So this is the idea that the regional species pool is filtered through to finer scale areas by both abiotic and biotic factors. And while these processes seem linear here, they're actually simultaneously occurring. They occur at the same time and they affect one another. So local species pools depend also on the dispersal ability of species at larger scales, not just the interactions between these filters. And invasive species in general act as part of the biotic filter largely, and they can alter the rules of species assembly. So moving on to that idea, we have this temporal idea of succession within certain kinds of ecosystems where you can have this alteration of an ecological community over time, species being replaced by other species or having differences in relative abundances. And there's two different kinds of succession that we typically talk about. One is primary succession, where you actually start over from ground zero. You have no vegetation, no soil. You're starting from the ground. And these are typically marked often by acute, intense disturbances that wipe out all the vegetation and other biota in the area. On the other hand, one that's considered to be much more common is secondary succession, where you have more often milder and more frequent disturbances that can change the trajectory of communities to different variants of final, what you'd say, climax communities if they ever get to that point. And then the different types of succession are, again, determined by types as well as intensities of these disturbances. So we're here to talk about exotic invasions, and as an overview, this is kind of a really old number, but even over 10 years ago, they were costing hundreds of billions of dollars annually within the United States worth of cost due to damages both ecologically and financially, and they threaten native biodiversity as well as ecosystem stability and function. And then also not only worrying about our native ecosystems, they also are threats to our agricultural systems as well. And then finally, there's a lot of different numerous hypotheses as to what creates a successful invader. And I highlighted three different things here that our lab has worked on in the past. Recently, Microstegium, as you just heard talk about, Acranthes japonica, Japanese chaflower, as well as Palmer amaranth. So, and also Erin did her work in the lab as well, so we did Lespedeza too. So this is just kind of showing like all these, just a small subset of hypotheses that actually are used to describe successful invasion. And this is a network analysis that shows that overall, based on these 33 hypotheses, there's roughly about 3 different neighborhoods that a bunch of these hypotheses fall under, and the size of these points indicate their relative importance as well based on whether other hypotheses reference these hypotheses in which they're built off of. And so you get this where you have 3 large clusters, but specifically if we're talking about disturbance, we're looking at this large node which looks at the invasion success of non-native species is greater in areas of higher disturbance levels. But another hypothesis that I was interested in for my research as well was the idea of the empty niche hypothesis, which is this idea that there is available functional hyperspace within these communities, and therefore if a job's not being filled, they can swoop in and make colonizations. So it's this question about, okay, what is an empty niche? This idea of a niche is very abstract, but there are ways that we can potentially quantify it if we look at from somewhat of a multivariate mathematical perspective, where you look at the lack of community saturation and overall species redundancy. So more species that are more similar in environment, they fill similar functional roles. The more roles that are filled, the greater saturation, the less niches that are available to be filled, right? Okay, so quite a few papers recently have been looking at ways to quantify saturation, and a really popular metric that's shown to have some good predictive power is 1 minus the mean pairwise distances, where there's if there's a greater of this number, it means that there's less niche availability and that the environment is more saturated based on the species that are there. So NPD can be sensitive to changes in distinct distinct taxa based on dendrograms or phylogenies, like evolutionary relationships. But it's important to understand that other than just phylogenetic relationships, you can also quantify and build dendrograms based on functional traits, things that you can actually measure on an individual plant other than just its nucleotide sequences. For this talk, I'm just focusing on the evolutionary stuff for that. So I had three major questions with the research. Number one, I wanted to understand if different types of disturbances affect a community additively or independently, or whether or not they interactively can change the trajectory of a certain assemblage of species. And then I wanted to understand how do exotic invasives, specifically exotic invasives, niches change in response to these disturbances over time, whether or not they become more or less prevalent during different points in succession. And then finally, I wanted to understand whether these communities that I'll be talking about are niche limited based on exotic or overall species richness, which is the number of taxa that occur in an area. So this is the experimental design that we had set up. This is in Touch of Nature Environmental Center in Makanda, which is just like a stone's throw away from SIU in Carbondale. And what you're seeing here is a split-by-split block design where we have different combinations of different fertilizer and mowing treatments. And I circled block 5 here because I actually have some nice pictures to show, as far as 2018, what these look like. These have been in progress since, we have data from up to 1996. So these have been in progression for over 22 years now. So you can see here that this is showing the different mowing treatments where obviously the control over 22 years has grown more woody. Somewhere in the middle you have 2 mowings during the spring and the fall, and then finally the spring mowing where it's a little bit more grown up, but it's not nearly as bad as our control. And then so for the fertilizer treatments that we did, our second source of disturbance, we actually did different kinds of nitrogen fertilizer treatments where you can see that back row. This is the same block, just on a different side, where that back row is the woody vegetation, but these are where those blocks occur this way. So you get 9 combinations of treatments for both different intensities of both different types of treatments, or disturbances. So that was kind of a picture that showed what it looked like when it was in full grown, when we actually took our vegetation and surveys. But this is what the treatments look like in fall. As you can see, only 1 treatment actually gets mowed, whereas the other 2 don't get mowed during that time period. So the methods that I used to actually do this is we actually collected abundance data of vascular species that occurred within each of these combination treatment blocks. And we assigned them cover class values based on what's called a modified Daubenmeier scale, which takes size class data and turns it into an arc-sine transformation based on their abundance. And so with that, we basically generated a dissimilarity matrix and created a non-metric-multidimensional scaling ordination, which showed these individual sites across years, and the closer that they are together based on these, the more similar they are in their species composition. And then finally, well, not finally, but we did a repeated measures PERMANOVA to test for differences in overall sites based on their composition, as well as a PERMDISP, which is a test for the homogeneity of dispersion, to see if within different combinations of treatments, whether or not certain areas had greater overall differences within their treatments. So did some combinations of disturbances basically allow for a larger variation in different kinds of communities? And then there was a pairwise-Adonis, which is basically a pairwise test to test different combination of treatments based on the PERMANOVA. And then finally, an indicator species analysis, which is shown in some studies to actually be a pretty good predictor of the characterization of a niche for a specific species. We did this for all species, not just exotic invasives, but we'll highlight the invasives here. So moving on, in order to actually calculate NPD, you actually have to build a phylogenetic tree. So we took the abundance data that was compiled from the 9 surveys throughout from 1996 up till current, and we combined them into a data set. And then for each of those, we went onto GenBank and actually pulled nucleotide sequences for both RBCL and MACK plastids. And so with that, we did an alignment, adjustment, trimming, concatenation, and then we actually built the tree based on inference of muscle, and we picked the best model to get the best tree with the lowest amount of uncertainty. And then we finally calculated our saturation index and then performed a regression analysis based on both the saturation as well and total and exotic species richness. So looking at the results here just from the NMDS, this is actually a 3-dimensional NMDS, but I feel only showing like this one panel of it is good enough because the other ones are fairly redundant in what they say. So this will give you the information you need. What this is showing is basically what these points are the observations of different sites over those 9 surveys. And what these eggs here kind of show, these are actually 95% confidence ellipses, which when you see like bar graphs and you see like there's a line that kind of characterizes the variation, that's what these eggs are doing. But the color of these are based on the different 3 levels of treatments for a certain set of disturbances. So you can see for the fertilizer treatments here, the control and then the 2 other fertilizers, you can see that they overlap quite well in the ordination space. And so therefore their location within the ordination is not significantly different. But you can see based on the differences in colors, they do have differences in their dispersion. Some of those treatments are wider or smaller, and the widest one here is actually the yearly fertilization treatment. So yearly fertilization promotes greater beta diversity or community dissimilarity within these sites. So looking at mowing treatments now, you can see that this large brown orange egg here is actually the control treatment for the mowing. This is where woody species can accumulate. And you can also see that it's also quite larger compared to right here where we actually have the other 2 mowing treatments that completely overlap not only in size, but they also overlap in location. So you can see that the lack of mowing causes greater dispersion within communities as well as a change in location. So then the lines here, these are actually vectors that were significant. And what they do is these trajectories explain the greatest rate in change for a certain variable, where this is total species richness, whereas this is the year. So as you can see over time, these 2 things have opposing trajectories, meaning that over time, there has been a decrease in taxonomic species richness for all species. However, the relationship between year, time, and this and exotic species richness, there's not really a clear trend. It's almost perpendicular. So it's showing that over time there is a decrease in taxonomic species richness, but it doesn't necessarily say there's a direct change related to time or taxonomic species richness for exotic species richness. It's not controlling the total number of exotic species that actually occur within a specific location. So the results for the PERMANOVA showed that instead of having singular significant effects based on different types of disturbances, it actually shows that there is some kind of significant interactive effect between different intensities of mowing and fertilizer treatments, not only based on the overall composition, but also the dispersion of different treatment types of communities. So, and then when you actually break that apart and talk about the pairwise, almost over 2/3 of these different pairwise combinations are significantly different from one another. It's not just like one pair of things is significantly different and throws off the overall test. All these things are significantly different from each other, save, 8 pairwise treatments. So after that, looking at the indicator species analysis, we had originally, from the 9 years of the survey treatments, we had 110 observed species within these plots that were established. And at some point in time over the 22 years, those 9 surveys, we actually had a large amount of them become what is considered a significant indicator for at least one regimen of a certain treatment at a specific time. And out of all those, 6 specific ones were actually exotic invasives, which I'll go through here in a minute based on this center for their criteria. But if you actually look at the pamphlet that you got today, it pretty well highlights the same species I'll talk about. So, and a lot of these you've actually already seen. So, for example, Slash orbiculatus, which is our oriental bittersweet, Slash-Diaceae family, is a significant indicator for the second to last treatment, basically the second to last survey, where it wasn't a positive indicator for mowing control, which kind of makes sense. It's a woody species. You're trying to mow it down. It doesn't occur there, so it makes sense that it would pop up in the control plot. So next thing, autumn olive here in its own family. It actually popped up relatively early in the surveys, and once again lasted through 2013, but dropped out in 2018 for some reason, and it was a significant indicator for the mowing control treatment as well. However, for Lespedeza, it was kind of interesting. This is the same species that Aaron talked about, where we actually saw it become an indicator, where we saw it actually go from an indicator for one set of disturbance regimes for a different one. It actually changed from originally when it was in the fertilizer control treatment, so it wasn't receiving any kind of fertilizer. And then in 2001, it actually changed over to being an indicator for a mowing treatment, which was the mowing for the spring and fall, which is kind of an odd shift there. And then Lonoceros japonica, the Japanese honeysuckle. It's been around since like the 2000s as a significant indicator, again, for a woody treatment, as one would expect. And then finally, well, not finally, there's one more after this, but Microcesium verminium, which we just talked about, it popped up just recently in 2018 in the mowing spring mowing treatment that we had. But the odd thing about Microcesium compared to some of these other ones is it has kind of like a triple whammy when it comes to being an invasive. It has open flowers for outcrossing, closed flowers for basically self-sexing, and then it also has this vegetative mechanism where if you cut it up, it can actually fragmentally pop back up. So when you try and mow it, you hit the possum and 2 more come up. So, and then finally, Multiflora rose here was only an indicator species in 2013 for the mowing control treatments. So that's just showing what it was only a significant indicator for that 1 year, oddly. So moving on to the phylogenetic tree. Again, we looked at 110 species. 3 of these species which created part of the root for this tree were the basal angiosperms, which are things like magnolias. And then the monocots here are in red, which were 29 of those. And then 78 eudicots over here. You have over here, like these are all the Asteraceae species, and over here you have a lot of your faboids. And this total tree length indice doesn't actually really have a metric because it's actually based off of substitution rates based off of nucleotide sequences, but some people care about that number. So finally, after calculating the near pairwise index for these, there is actually not a significant relationship based on exotic species richness. As you can see here, most of the plots actually have just one exotic species within them. But taxonomic species richness has somewhat of a different story, where overall taxonomic species richness actually has somewhat of a negative relationship with saturation. And you think about that, and it kind of is backwards from what we originally talked about. And what I we'll be looking at soon in my research is I think the reason we have this is because there are exotic species that are dominating a large portion of these sites over 22 years. So what I want to do next in my analysis is with these linear regressions, I want to include these disturbance treatments as cofactors to see if those are the things that are actually driving this regression. So in conclusion, it shows that the composition and the dispersion are based on the interactive effects of separate disturbances that are happening at the same time. And then exotic species actually have increased as indicator species, but largely mostly for the control plots. Although we did see variation within these, so it's really important when we go to specific sites for management, it's important that we manage for all the species there and come up with a several-step plan to deal with multiple different exotics that have different functional relationships with the environment. And then finally, something that I forgot to bring up is that the only treatments that didn't have any exotic invasives as indicators were the 5-year and the annual fertilizer treatments. Those two treatments, they had exotic species, but they were not exotic, but they were not significant indicators for those treatments. And then finally, based on what we found out from the linear regression, is that all species niches were limited within the community because of dominance. And however, we're seeing that exotics are still being able to invade these areas regardless because they become more commonly significant indicators over time. And then finally, what we're doing is we're going to continue the consistent maintenance of these areas and resurvey them in several years to come to see how further long-term changes affect basically how longer periods of time show differences in these relationships. So with that, I would like to thank all of these people who have contributed either by helping maintain the plots, they put up previous research, actually doing some of the fertilizers treatment with Shen in Jersey. Here, our funding source, and that's the National Science Foundation, Touch of Nature for helping with the mowing treatments, and then finally, SIU Plant Biology. And with that, I can take any questions.
Are these plots in Carbondale only, or are they in other sections?
It's just one site in the Touch of Nature Environmental Center in Carbondale. I would say about, I'd say one of the, so let's go back to these are not very big areas at all, but these individual sites are probably about 2 or 3 meters by 2 or 3 meters for individual treatment combinations.
It seems to me that, it's kind of over my head, but it seems to me that the dynamics for annuals would be very different from the dynamics for non-annuals in terms of niche engagements over this period.
Yes, we're planning to expand this analysis to include not only functional groups, but also functional diversity as well, where we're going to account for annual versus perennial life history traits. So that is the next part of the analysis, along with the basically fine-tuning the regression analysis. This is stuff I literally just did 2 days ago. So sorry about that. Yeah, it was like, make sure I get it right. Sorry.
How much longer till you get your PhD?
4 years.
Oh my God.
This is a fellowship project. This isn't even actually dissertation stuff. So it's a different thing. Thanks, guys.